Hello everybody, in this video I'm going to be discussing the AQA A Level Biology topic nerve impulses. So on your specification this is section 3.6.2.1. So in this section of the specification we will discuss the structure of different types of neuron, specifically the myelinated motor neuron, but I'll go through others for context. And then we'll talk about how the resting potential gets established, how an action potential is formed, and different aspects of how that action potential is conducted along the length of the neuron. So to begin with, here we can see the structure of the three major types of neuron that we find in the nervous system. So this first type of cell here, drawn in black, is the sensory neuron. Now the sensory neuron connects receptors to the central nervous system. So the receptor would be over here. These little narrow extensions coming out of the cell are called dendrites, and they form connections to other cells. And then we've got the extension here heading towards the cell body, which is called a dendron. Coming out of the cell body and onwards is the axon. So this here is the cell body, where the nucleus is, and also mitochondria and other organelles necessary for maintaining the function of the neuron. Here we can see the myelin sheath, which is a series of related cells called the Schwann cells, which produce this myelin sheath around the outside, and I'll discuss that more later on in the video. Over here you can see that there are gaps in the myelin sheath, and one of those gaps would be called a node of Ranvier. This green cell here is a relay neuron, sometimes referred to as an intermediate neuron, and these are found within the central nervous system, and they connect sensory neurons to motor neurons essentially. Sometimes there's multiple relays though, so they're in the central nervous system. Their job is to connect the right sensory inputs from sensory neurons to the correct motor neurons that are going to lead to the effectors to carry out the responses. So again, here we see that it has a cell body with the nucleus, etc., and it also has dendrites extending out of it to form connections to other cells. Now this purple cell that it's connected to over here is a motor neuron. And motor neurons connect the central nervous system, or the relay neurons specifically, to the effectors. So effectors are things like muscles and glands, and they produce the response. So again, we can see the cell body with dendrites, and this time we've just got the axon extending out of it. So here is the axon, and again there is a myelin sheath which is being produced by the Schwann cells and nodes of Ranvier in between them. At this end we're going to have the effectors. So you might have, for example, if this is connected to a muscle, a neuromuscular junction, which is covered in a different topic. And I also in this video won't be talking about synapses, which occur here and here between neighbouring neurons. That again is covered in a different part of the specification. So when you looked at nervous control of the body, you will have learned about reflex arcs, which is the simplest arrangement of just three of these cells. So you get your stimulus, which is detected by some sort of receptor, and then a sensory neuron carries it to the central nervous system, where it goes to a relay neuron. That's passed on to the correct motor neuron, which then carries the impulse to an effector, which carries out the response. So this is the process of stimulus to response in a simple reflex arc. So SR, SR, MER will help you remember all of the different steps in the correct order. Now the first state we're going to look at with regards to this neuron is called the resting potential. The resting potential is when the nerve is not carrying any impulses along it. So this neuron has a polarized membrane at this stage. Now a polarized membrane indicates that there is more charge on one side of the membrane than on the other. So in this case, it's more negatively charged inside the axon than it is outside of the axon in the tissue fluid. So within the membrane of our neuron, we have several different types of channel proteins and carrier proteins. And these carry positively charged ions, so sodium ions and potassium ions. Now, during the resting potential, the sodium gated channels are closed, so any sodium ions that are outside cannot enter into the axon. Now, why would there be so many sodium ions outside? Well, there is an active transport protein embedded within the membrane called the sodium-potassium pump, and this is the primary driving force behind the establishment of this resting potential. So the sodium-potassium pump takes 3 sodium ions from inside the cell and pumps them outside. At the same time, it takes 2 potassium ions and brings them into the axon. Okay, so by taking in 2 potassium ions, which are positively charged, but removing 3 sodium ions, which are also positively charged, there is a net loss of positive charge from the inside of the axon, from the cytoplasm of the axon. So that is going to reduce the membrane potential. It's going to make it more negative. In addition, this sets up various concentration gradients. So that outside there's going to be a high concentration of these positively charged sodium ions. However, as I mentioned before, the sodium channels that they could re-enter the cell by, down their concentration gradient by facilitated diffusion, are closed, so they won't be able to re-enter the cell. Now the potassium ions that we've pumped into the cell become in a high concentration inside the cell, and some of them, not all of them, but some of them will be able to essentially leak out because we have some open potassium channels, and that obviously is also going to contribute to the increased polarity of this membrane, so it's going to become more and more negative inside. Within the neuron, there are also some permanently negatively charged ions and proteins and other molecules which contribute to the negative charge. Now the resting potential itself, in terms of quantifying that, is going to be minus 70 millivolts. So there is more positive charge outside of the cell than inside of the cell. So now that we've established the resting potential within the neuron, we're now going to look at how it can carry a nerve impulse. And the nerve impulse is in the form of what's called the action potential. Now on this graph we can see the progression of the action potential in terms of that potential difference across the membrane. At the beginning of the graph here, we see the resting potential that we just established, minus 70 millivolts. Here is the beginning of our action potential. The first phase is called depolarization. Now when we established the resting potential, we said that the resting potential is described as polarized. The membrane is polarized. Okay. In depolarization, we are getting rid of that negative potential difference across the membrane, and it actually becomes slightly positive. It goes up to about plus 40 millivolts. Now the reason for the depolarization is because we're going to take lots of positively charged sodium ions into the cell. After that initial depolarization, there is a change in the membrane that we'll look at in a minute, and we get repolarization, which is a process of becoming back to that negative state again, and that's because of removing potassium ions from the cell. So both sodium and potassium are positively charged. So if we have positive charges coming in, we're going to become more positive, and if we've got positive charges going out, we become more negative. There's going to be a short period of time here where the membrane is even more polarized than it was before, so it might dip down to about -80 or so, and that's called hyperpolarization. And then we'll return to the resting potential again. So that is the process of one single action potential in terms of the charge across the membrane. Now we'll look at how each of those stages is going to occur. So when you have what we call the threshold potential exceeded, which we'll look at in a minute, we initiate a process called depolarization. So the energy to start this comes from a change in the potential across the membrane. In order to depolarize the membrane, the voltage-gated sodium channels that we talked about before open up. And because we've got this large concentration of sodium outside the cell because of the sodium-potassium pump in the resting potential, once these channels are open, lots of sodium can enter. Now, when we have the initial depolarization or initial stimulus placed onto this neuron, only some of these voltage-gated sodium channels will open. So we get the membrane potential starts to increase from that -70. If it reaches a threshold, then more of them open and even more sodium comes in. Now that threshold that it needs to reach is when it gets to about -50 millivolts. Once we've got beyond that, more and more sodium channels open, and we get that massive influx of sodium ions, and we get the increase in the membrane potential up to about +40 millivolts. So this is the depolarization stage of the action potential. An important aspect of the depolarization stage of the action potential is understanding the all or nothing principle. The all or nothing principle tells us that if we don't reach this threshold, we won't get the full action potential. We won't have an action potential at all. So at about -50 or -55 millivolts, we've got a threshold that the cell needs to get to become depolarized. So if we get a very small stimulus, or small kind of electrical stimulation of the membrane, some of those voltage-gated sodium ion channels will open. But it won't be enough to reach the threshold, so this is called a failed initiation. We haven't met the threshold. If we get slightly more of those sodium ion channels opening, voltage-gated channels, still not enough to meet the threshold. But if we get a stimulus which is big enough to break past that threshold, we get the full response. It goes all the way then. It opens enough channels to go to a full depolarization. And then we get the complete action potential. So this is the all and this is the nothing. If you meet the threshold, you get an action potential, and all action potentials are the same size. They all go up to about +40 millivolts. But if we don't meet the threshold, we get absolutely nothing. We don't get an action potential, so it fails to initiate the action potential. So as a result of this, all action potentials are the same size, regardless of the stimulus, and they all last only about 3 milliseconds. Once we've depolarized the membrane, then a repolarization occurs. Because we don't want the action potential to last indefinitely, we want to be able to send another action potential after it. So it's going to repolarize back down to that resting potential. Now this is caused by a decrease in the permeability for sodium. So after about half a millisecond of depolarization, the voltage-gated sodium channels close. So the influx of sodium ions is going to stop. The voltage-gated potassium channels are open. So potassium ions from inside the axon move out down their both chemical and electrical gradients. So this takes with it obviously positive charge because potassium ions are positively charged. So this is reducing the membrane potential. It's getting more and more negative back down towards the resting potential. We're also going to see the sodium-potassium pump kick in again. So the sodium-potassium pump begins as it did before, actively transporting sodium ions out and potassium ions in to the axon in a ratio of 3 to 2. So again, this is removing net positive charge because it's removing more positive ions than it's bringing in. So once we've initiated the action potential, we get a wave of depolarization along the membrane. This means that once we've begun an action potential, it will continue along the neuron on its own. So here we can see the resting potential. A higher concentration of positively charged ions outside of the neuron than inside the membrane. This is the point of depolarization. The flipping of the charge across the membrane, now positively charged inside. So here's our action potential. And we've got then the repolarization, which occurs a tiny fraction of a second later. Here the membrane is in the recovery period, where it's getting ready to send the next action potential along behind. And all that will happen now is that this depolarization here at the front of this wave, that provides the energy needed to open the voltage gated sodium channels in this part of the membrane here. And then positive charge will move in and this will flip to being positively charged inside and negative outside, and the whole thing's going to continue along. This also matches up with our graph of time, right? So the resting potential -70, going up, depolarizing, coming down, repolarizing, slightly hyperpolarized for a bit, and then we go back to the resting potential. All the same stages matching up on these two diagrams. Now the type of neuron that we focus on most in AQA biology is called a myelinated motor neuron. And myelinated refers to the presence of the myelin sheath that we looked at in the structure of the neuron at the very start of this video. Now the myelin sheath is actually produced by a different cell, not the neuron itself. Here we can see over on this diagram a Schwann cell. The Schwann cell, with the nucleus here, wraps itself around the axon, that long extension of the neuron. And the membrane is lipid rich and it contains this myelin. The myelin sheath insulates the neuron electrically. So as a result, not the entire membrane needs to be depolarized. So when we are conducting the action potential along this neuron in this direction, as that wave of depolarization reaches here, and this becomes depolarized, so positively charged inside and negative outside, this next section with the Schwann cell around it and the myelin sheath around it is electrically insulated, so the action potential essentially jumps to the next node of Ranvier. Remember, these are called nodes of Ranvier between the Schwann cells. So once we've depolarized this, the action potential jumps to here. So what we don't need to do is depolarize the full length of the axon membrane. And because it's skipping steps like this, the conduction is much faster. So this type of conduction is called saltatory conduction. And it means that the action potential jumps between nodes of Ranvier for faster neuron conduction. Now, myelination is not the only factor which affects the rate of conductance along a neuron. Other factors that affect it are the diameter of the axon and also the temperature. Now, the diameter of the axon affects the surface area to volume ratio of the axon. So if we've got a larger diameter for the axon, it's actually a smaller surface area to volume ratio. So the membranes sometimes leak ions through them. And more leakage of the ions means it's harder to depolarize the membrane, because we can't control the movement of the ions in and out as efficiently. So with a larger diameter, we get less leakage of ions and faster transmission of the action potential along the neuron. Now, temperature affects the conductance along the axon in two ways. At higher temperatures, we have more kinetic energy within the particles. That's true in all cases. Now, in this situation, why that's important is that we're relying on the diffusion of the ions in and out of the cell. When we open up voltage-gated sodium channels or voltage-gated potassium channels, those ions diffuse through by facilitated diffusion. So at higher temperatures, you get faster facilitated diffusion. And another factor is that there are enzymes involved in the process. The sodium-potassium pump is controlled by enzymes, so the sodium-potassium pump is more efficient at higher temperatures, as long as the proteins haven't been denatured. So we're not talking about very high temperatures. Now, earlier I mentioned this phase of the action potential called hyperpolarization. Hyperpolarization is when the membrane potential temporarily dips below -70 following the repolarization stage. Now, when the membrane is hyperpolarized, the membrane is less permeable to the sodium and potassium ions. So if we kind of zoom in on this phase of the hyperpolarization as it's returning back up to the -70, there is a period of time immediately after the previous action potential where you absolutely cannot send another action potential along this neuron. That's called the absolute refractory period. So membrane permeability here is 0. And as the membrane potential returns back to -70, the resting potential, it becomes easier and easier to send another action potential after it. But just after the absolute refractory period, at the start of the relative refractory period, it's harder to send the action potential than it is here when it's closer towards the resting potential. Now, the refractory period is important in making the action potentials discrete events. We don't have an action potential that lasts indefinitely. So this is not a switching on and off of the neuron, it is sending one nerve impulse down the neuron towards the next cell. And that allows us to communicate the strength of the stimulus along the axon. So when we have a very weak stimulus from the previous cell, our neuron will not meet the threshold. And as a result of not meeting that threshold voltage across the membrane, there will be no action potentials. In response to a weak stimulus, the threshold can be met, but not very often. So we get a very low frequency of action potentials along the membrane. If you've got a slightly stronger stimulus coming along the axon, then we get more frequent action potentials. But then for a very strong stimulus, as soon as that refractory period is over, we're basically sending the highest frequency that we can of action potentials along this neuron. So this is how the strength of stimulus is communicated within the nervous system. It's all about the frequency of the action potentials. But notice here that the action potentials are all the same size. Remember, the all or nothing principle tells us that once we meet the threshold, we produce an action potential of the same size. So returning to the specification again, you can see what you need to be aware of for this topic. You need to know the structure of that myelinated motor neuron, how we establish the resting potential, but then how we have an action potential transmitted along that as well, and the various different ways in which the conductance of that action potential is affected. And just to finish off, we're going to have a look at some questions so you can test your understanding. So have a little read of these questions, pause the video, try them, and then come back and I will talk you through how you could get maximum marks on these sorts of questions. So looking at question 1, it asked us to describe how the resting potential is established. So in an exam when we're asked to describe, we are simply going through the steps of what occurs. So things that you should have mentioned in your answer are the sodium-potassium pump. And then you also, when you're discussing the sodium-potassium pump, you need to be saying what it's doing and the ratio of the ions that it's moving. So if you mentioned in your answer that you've got 3 sodium ions being pumped out for every 2 potassium ions that are pumped in, that would gain you an extra mark. The next thing to mention is that some of the potassium ion channels are open, so some potassium diffuses out or leaks out. And you could also get a mark for describing the kind of end result of all of that, which is that situation where we've got a minus 70 millivolt potential difference across the membrane. So 3 of those 4 things would get you 3 marks on that answer. So question 2 was an explain question. So we're going to be looking for the core idea behind the concept when we're looking at explain questions. And the main idea with the all or nothing principle is that you have to exceed the threshold in order to produce the action potential. So ways you could express that idea in your answer would be statements like the stimulus must open enough sodium channels to exceed the threshold in order for an action potential to be generated, or changes in the membrane potential which exceed the threshold lead to an action potential. If it don't reach the threshold, then we don't get an action potential. So that's one idea that needs to be in there. And the second thing is that there is the idea of this kind of all or nothing. And what that means in relation to all is that all action potentials are the same magnitude. Don't necessarily need to use the word magnitude, but the same size, the same change, or you could say they all go to plus 40 millivolts. Yeah. So this idea that it's always that shape, it's always that size, from minus 70 up to plus 40 every single time. A bigger stimulus does not mean we go higher than plus 40. Small stimulus doesn't mean we go below plus 40 unless it's not enough to meet that threshold. So it's all or nothing. So question 3 asked us, why do action potentials propagate faster along myelinated neurons than non-myelinated neurons? So remember, our myelinated neuron has the myelin sheath and then nodes of Ranvier in between the areas of the sheath. And the action potential is going to jump from node of Ranvier to node of Ranvier as it's being propagated along. Whereas in the non-myelinated neuron, it needs to go all the way along the membrane. So in order to get marks on this, we want to describe the situation. Whenever you're basically doing a comparison question, you must remember to talk about both sides. Don't just imply what happens in the other. So we would explicitly state that in the myelinated, the action potential jumps between nodes of Ranvier. And it's always important when you've got a concept like this to mention the name of that concept. So give yourself another mark if you said that this is called saltatory conduction. And then we need to address the other side of the situation. So the non-myelinated neuron, the entire length of the axon membrane must be depolarized. So it's almost like the myelinated neuron is taking a shortcut. It's traveling a shorter distance because it's depolarizing less of the membrane. It's simply doing the gaps between the myelin sheath. Okay, hopefully this video was helpful to you in your revision of this topic. Thanks for watching.